Hello everybody. In the previous video we have studied what is a plant disease, what are the important terminologies in plant disease cycle and the classification. In this video you're going to study few of the plant disease. So I have picked 4 plant diseases which we will study in detail about them. So what is the necessity of studying plant diseases? We know the plant is the only producers on planet Earth. Since they are the only producers, we have to know what are the pathogens attacking it, how can we protect it and have healthier seeds or healthier yield from it. So we are going to study the plant diseases so that we will know which is the pathogen attacking it, what is the symptoms. By looking at the symptoms, we will be able to take some of the controlling measures or precautions from getting the plant completely rotten by the pathogen. So first disease which I have taken up for this class is Ticca disease of groundnut. So Ticca disease, you can say that it is the scientific name of groundnut is Arachis hypogea. Okay. So these have 2 causal organisms. One is Cercospora aradicola, the other one is Cercospora personata. So these are the 2 different causal organisms, that is species of organism which is causing disease in groundnut. So Ticca, as the word says Ticca, it is the spot. So this disease is specially spotting of the leaf. Okay. So now when we get into the classification of the causal organism, the class Deuteromycetes, order Monililiaceae and family Dematiaceae. So the introduction for this disease is there are nearly 3,800 of species which is causing these disease. Now what are the speciality? So as I said, it is also called as spot disease. The leaf will carry brown spotting. Okay, so we say it as spot disease. So this is a parasite which is very common in groundnut, cotton, pulses, etc. Now moving to the vegetative structure. So how is the structure of these causal organisms? So there is variation. Since there is 2 different species, we will have 2 types of structures. One is for personata and the other one is aradicola. So in the case of personata, you can see that the mycelium is highly branched, septate, and also they are going to grow in the intercellular spaces. Whereas in aradicola, you can see that they have conidia which is called as obclavate, that is long obclavate spores. So conidia spores obclavate in the sense they are club shaped and you can find one having an opposite direction. So that is called obclavate. And then they are hyaline transparent in nature with about 4 to 12 septate. Now symptoms. The first symptoms is seen in the aerial part. So the entire aerial part is going to show the symptoms. So what is the symptoms of personata? Personata will show spotting of the leaf which will be circular, pale in color and then change into dark brown to black. Whereas in the aradicola, the spots will be circular at the beginning and later on they become irregular. Okay, so here you can see aradicola. Then you can see the coloration also changes. The color is changing from reddish brown to black in color. So how is the spotting seen? So the leaf, okay, if that's the twig, they'll have the leaf which shows a circular spots. So that's the reason we call it as Ticca disease of groundnut. They have circular spotting on the leaf. So the surface, that is ventral surface, will have maximum spots, whereas the dorsal surface, that is the lower surface, will have few spotting there. Then moving on to disease cycle. So they say that the fungi is being present on the debris, that is the shells of the groundnut, the root, the previous year's harvest. So the remaining waste which is present, that is the place where the inoculum is present. Inoculum in the sense the fungi mycelium is present. So how is it being transferred? This mycelium can be carried with the wind, or even the conidia can be carried along with the wind. So this conidia, when they fall on the same groundnut plant or a different groundnut plant, they start to put forward the germination tube. So from the conidia, there will be a small germination tube coming out. This germination tube can enter through the free entry, that is stomata, or if they are falling on the upper surface, that is the ventral surface, there is all chances that they can pierce the epidermal cells and then proliferate. Now, so once they get inside, so what is the conducive climatic condition? They require warm and moist condition. So it is about 24 to 28 degree temperature with high humidity of 3 days will give a very good incubation period for this particular species to grow in groundnut. Now, how is the infection occurring? It's again the same way. The infection is by the conidia which is falling onto the leaf surface. They will pierce the epidermal cell. The germination tube goes inside. It moves intracellular and then they aggregate the mycelium. Aggregation is the mycelium come together, they fuse together, then they give rise to conidiophores. Okay, this conidiophores are special structures on which conidia has been born, or it's bearing conidias. So this conidias can be, okay, so this conidiophores will give out conidia through the stomata, or rather through the epidermal cells. Again, they can pierce out the epidermal cells and the conidia can be, conidiophores comes out and then the conidias is produced on the conidiophores. Now, so the infection, how is the infection occurring? It can be on the same plant or different plant. So if the wind carries conidia from the aerial apical tip of the plant is fallen to the somewhere basal part of the leaves, then again there will be secondary infection occurring but on the same plant. At the same time, they can allow the infection, they can infect the healthy plants around it. Okay. Next, what is the control measures? So the first thing is burn the debris. So why burn the debris? The previous year's yield, the shells, etc. When they are burned, the mycelium is destroyed. The next one is seed dressing. So what is Virivax? It is a particular chemical about 2 grams per kg of the seed can be covered with Virivax, okay, and that will prevent the mycelium from growing. The next one is foliate spray. So Bryophytes mixture, that is the best fungal fungicides which is available. So that mixture can be taken and sprayed on the leaf to prevent the spread of ticca disease. Okay, so that is about the ticca disease of groundnut. The second disease which we are picking up for this class is late blight of potato. So which is the host? Phytophthora infestans. Okay. Causal organism, Solanum tuberosum. Okay, so what is the classification? They belong to the class Oomycetes, order Peronosporales, family Pythiaceae, genus Phytophthora. So this particular late blight of potato was first seen in North America in the year 1845. Okay, in India it was first spotted in the Nilgiris. Okay, somewhere around 1870 to 1880. We are not very correct about which year it is. Okay. So then after that it is seen in all the hilly regions. Okay. And it is worldwide spread. Now moving to the causal organism, it is a parasitic. Okay. Aseptate, they do not have septa. When we say they are aseptate, it is understood that there are plenty of nuclei in that particular hyphae. So they are cenocytic, they are branched and they are intercellular or intracellular. Here the movement of mycelium is both inter and intracellular, through which they are going to produce hostorias, and this hostorias will be extracting the nutrient from the host cells. Okay. Now, talking about the disease cycle, again they require high humidity and moisture content. Now, when we say moisture, it is even for the tubers, even in the soil they require moisture. Now, what happens? The wind will carry the spores, that is zoospores, and they are going to help in spreading the disease. So, the temperature is about 21 to 24 degree centigrade. Now, in this case, whereas in the case of potato, that is the tubers, what are the symptoms? You can see that in the case of tubers, we say that the tubers consist of nodal region from which we are going to get new plantlet. But whereas this mycelium will grow completely inside the tuber. So that's how they are going to penetrate in the tuber and they will infect or infest the new plantlet. Now, moving on to symptoms, the first thing you can see is the margin of the leaf is curling off. Okay, so they have wavy kind of the leaf, and then you can see the petiole and also stem is carrying the spots. Okay, what kind of a spot? Brown spots. Okay, this spots will increase as the infection is going higher and higher. Now, so there is another special thing that is called as tuber rot. As I said earlier, the moisture content in the soil is also important. If the soil is having lots of moisture, then the tubers, that is the potato tubers, will become rotten. Rotten is watery in nature. Okay, they can become watery in nature. And they say that even the tubers will show brownish spots which will turn into purple color. Okay, so that is another symptoms of late blight of potato. Now, what are the disease management? How can we manage this? So there is again the Bryophytes mixture. Why is it? It is the best fungicides. That's reason almost all the fungal diseased plants can be saved by this Bryophytes mixture. Next, so the tubers should be from a disease-free place. Now, as I said, late blight of potato is worldwide distributed. Okay, so when we collect the seeds in the sense the tubers, they should be free from disease. From which region we are collecting the tubers, that region should not be having the history of late blight of potato. The next one is burn. So whenever we come to know that this particular plant or that particular tuber is being infested, it's best to burn that particular plant. Why is it so? The mycelium will not be further growing, but it will be killed completely. Next, to get a disease resistant variety. So that is the best option. And the last one is sanitation. Sanitation is clearing the field, the leaf which has fallen off or the previous year's yield, they should be cleaned, removed off from the field and also solarized. Solarized in the sense the field will be sanitized. Okay. So the third disease which we are talking about is citrus canker. So which is the host species? It is citrus species. Citrus species, it can be the lime, sweet lime, oranges, etc. So which is the pathogen? It is Xanthomonas citri. Xanthomonas citri is the pathogen. The class Gamoproteobacteria, then the order Xanthomonadales, then the family Xanthomonadaceae. Okay. Now, what is the introduction? Again, we know that this is the first bacterial disease which you are going to study. Okay, and it is worldwide spread. It was severely seen in India, Japan. Okay, so this is the places where it was seriously hit. Now, moving to the symptoms. Symptoms, scabby spots. Scabby is irregular, cracky and even rough kind of a spot. We call it as scabby spots. So we can see that is nothing but small cankers, a spot. Okay, if this is the line, okay, if this is the line, you can find that there will be spotting, brown spots on the lime, even lemon, and even on the leaf also you can find brown spots, which is primarily seen as a brown spot. After few days that particular spot will raise, okay, from the surface of the leaf or the fruit. You can find there is a slight increase in the spot, okay, region of spot. So that is called a slight raise. So where do you see these spotting? You can see them on leaf, fruit, young branches, okay. So these are the places where you can see the spotting. Moving to the causal organism, the structure, it is a short, rod, motile bacteria. It's a short one which is having a single flagellum, polar flagellum, okay, and they are aerobic in nature, and also the rod-shaped bacteria is surrounded by mucilage sheath, okay. So that is about the bacteria, which is a gram-negative bacteria. It requires mild temperature and wet condition. So this particular bacteria will flourishingly grow when the temperature is about 20 to 30 degree centigrade, okay. So the disease cycle, you can see that the inoculum or the means of spread is the old lesions. So whatever I said, the spotting which is seen on the citrus, that is the fruit, okay. So the fruit consists of some spots on them. So cankers, we call it as cankers. So in this cankers, some cankers are watery in nature, okay. So they are watery in nature. Some of them are dry, as I said, rough, scabby spot. So the one which is watery, they are the one which consists the bacterium. It is said that about one watery lesion may contain about 50,000 of rod-shaped bacteria. So these old lesion is the primary inoculum. So from that, from the lesions, from the old lesions, from the watery lesions, only the bacteria are spread. Now, how is the means of spread? It is by rain or man. So rain is the one which is spreading it. So once the rain is hitting this lesion, the rod-shaped bacteria is carried along with the rainwater, and when it falls on the healthy leaf or the healthy plant, it starts infecting. So human is also the one person, that is, we get the diseased seed and plant it. So it is the one which is causing the citrus canker to spread from one region to another region. Now, talking about control measures. So first one is pruning. Pruning is removing of the diseased leaf. So whenever we come in, whenever we see a diseased leaf, they are need to be pruned. So there is pruning scissors which has been used by the farmers to remove off the diseased part of the plant, okay. So the other one, eradication. Eradication is removing of the diseased tree from that particular spot. The next one, disease free stock. Disease free stock is wherein we are going to get the genetically modified plant which will resist itself against this Xanthomonas citri and grow flourishingly. The next one is a particular antibiotic. It is said that streptomycin sulfate, when it is sprayed to the citrus canker, that is the cankered regions of the plant, they are going to reduce the disease, okay. So that is about citrus canker. Next, moving to the fourth disease, that is the tobacco mosaic virus. Tobacco mosaic virus, the host is Nicotiana tabacum and the pathogen is tobacco mosaic virus. So looking into the classification, they belong to the class Alsoviricitis and order Martilvirales and family Virga Viridae, and then coming to the introduction. So introduction is for the first time Stanley was the person who crystallized this virus. Okay, so it was Iwansky who first showed that when the tobacco leaf extract was taken, okay, so when the leaves were crushed and the extract was taken, he showed that once this extract had passed through the bacterial proof filter, still they were showing some kind of symptoms on the healthy plant. Okay, so when he poured the extract of this infected plant onto a healthy plant, they said that still, even though it is passing through the bacterial proof filter, bacterial proof filters are those filters will have pores which is minute, which will not allow the bacteria to pass through. So Iwansky said that there must be some organism which is much smaller than the bacteria which is causing this disease. Okay, so that extract, in that particular extract, the virus can be viable for 25 years, okay, in that particular leaf extract. Now, Stanley then crystallized those compound and found out that the virus is the causal organism. Now, moving on to symptoms. The primary symptom is the chlorotic lesions. Okay, so you can find a small chlorotic, that is the death of chlorophyll. So that particular regions will start turning yellowish in color and later on become brown in color. Now, they say that in the secondary symptoms is the dark green spots which becomes irregular in shape, and then they are going to make that particular spot stays as such and the entire leaf will turn yellowish in color. Okay, that is chlorosis occurs. So this spots will crumpled blisters. Then you can see the crumpled is not a clean thing which has been just managed like that. Crumpled blisters and the entire leaf turns chlorotic. The first thing is they show systematic mosaic type. Okay, so the patches are mosaic types. Okay, you can find like those patches on the leaf of tobacco which shows curliness at the leaf margin. Okay, you have mid veins. Okay, so that's how you can see the symptoms. And there is one strange feature, that is there are different strains of viruses. So there are, based on the strains of viruses, the symptoms vary. If one of the strain of the virus shows chlorosis, the other strain will show only necrosis, no chlorosis. Then if one strain is showing spotting, another strain will show streaking. Okay, streaking is lines like this, streaks on the stems, etc. So like that, based on the species or the varieties of strain, the symptoms are seen. Now, talking about causal organism shows rod shape. That is, this particular tobacco mosaic virus will have RNA, genetic material as RNA, single strand RNA, which is surrounded by protein. Okay, so this protein consists of 2130 subunits in them. Okay, so that is about the causal organism. Now, moving to the control measures. So, it is said that this particular virus is being transferred, or the primary inoculum is the debris or the warehouses wherein they are discarding the tobacco which is utilized to make cigarettes and other tobacco based products. So when the warehouse is left out, that particular regions will have this virus. So they prefer the seed bed to be very far from warehouses. Tobacco warehouses, from very far the seed bed should be. And sterilization of seed bed should be done. So how is sterilization is done? It is like steaming method. Okay, so when the steam is passed over the seed bed and they are turned over, they are getting sterilized. Then the next one is burning. So whenever you come across the tobacco plant which has been infested with TMV virus, they say that to remove off the plant and burn it completely. The last one is the resistant variety. So when we get the resistant variety of plant which is resistant to TMV virus, then surely the plant can sustain from that particular diseases. So another small topic to be covered here, we will start with bryophytes general characteristics. So to begin with, bryophytes is derived from the Greek language bryon. Okay, bryon means moss. Okay, bryon means moss and phytes is nothing but plants. There are about 2,500 species of bryophytes and we say them as amphibians of plant kingdom. Why do we call them as amphibians? Even though they are land plants, with few exceptions like polytrichum, which is xeric condition, sphagnum, which is seen in bogs, and ricea, which is seen on substratum thalloid nature on the rocks. Okay, but still these plants require land and water for their survival. Okay, so water is very essential for fertilization's sake. And the other thing is, we can see that there is difference in the body structure. They can be thalloid or leafy in nature. Thalloid in the sense, you can see that they are dorsi-ventrally flattened and they are growing on the flat surface on the rock and they are compressed on both the surface. That's the reason we say dorsi-ventrally flattened surfaces. Now, the other thing is leafy ones. For this you can have Mercantia as an example, and for the leafy one we can take as Funaria as an example. You can see that there is a leaf-like appendages and also root-like structure which is called as rhizoid is present. Okay, and that's not the fruit, it is a sporophyte. We will know about it in detail. Now, the rhizoid is of three types. Either they can be smooth unicellular rhizoids, as in case of Mercantia, or you can see that they can be tubercelled. Tubercelled in the sense the outer surface is smooth and a single layer, whereas the inner surface you can see they are moving towards inside with projections. Okay, such kind of rhizoids is called as tuber-celled rhizoids. And the next, these are all unicellular. When we move on to the multicellular rhizoids, you can say that there are simple rhizoids with oblique walls. Okay, so they don't have a straight walls, it is always oblique walls was present in the case of multicellular rhizoids. Next, moving to the vegetative reproduction. In the case of vegetative reproduction, you can see there are 5 different types. The first one is adventitious branches. Now, when we say the plant has to grow, it grows in a conducive environment. That is, any time the plant will not survive luxuriantly, flourishingly growing. Okay, so only when the climatic condition is conducive, it is supporting the plant to grow, only then they grow. In case there is an unfavorable condition, these vegetative reproduction is the one which is helping to survive or perpetuate this particular bryophytes. So the first one is adventitious branches. The adventitious branches is when the thalli, okay, when the thalli is present, along with it laterally there will be small lateral branches growing. Okay. So these branches is the one which will persist itself when the climatic condition is changing, and they will grow into a new plantlet. The next one is death and decay. When the plant is getting older, that is the thaliform, thallus one which is getting older, what happens is the plant entirely dies and decays, but only the tip will survive. The tips which survive can grow into a new plant. The third one is persistent epiphysis. Persistent epiphysis is much similar to death and decay, but it is not due to the old plant, but it is for the unfavorable condition. When the unfavorable condition arises, the particular parts will disintegrate and only the tip will stay as such. So that is called persistent epiphysis, which will be growing into new plant. The next one is tubers. When the plant is growing, there is a chance of accumulation of the reserve food materials. When they accumulate at the tip, which is irregular in shape. Okay, they are irregular in shape, and when there is unfavorable condition, same thing, the entire thalli dies and decays itself, whereas the tuber will survive it and they will grow into a new plant. The last one is the gemma. Gemma is got when mercantia, it is seen in mercantia, wherein they grow a special structure. Okay, that is called a gemma cup, which will have plenty of gemmae inside. Okay, so plenty of gemmae inside, which is a stalk, which has a stalk and a upper portion which is broader and elongated one. So each gemma is capable of germinating and growing into a new individual. So that is about vegetative propagation seen in bryophytes. Moving to the sexual reproduction, it is by the production of antheridium, which is the male sex organ, and archegonium, which is the female sex organ. So how is this seen? The structure will have a cavity, which will be present in a cavity, which will have a roof. Okay, so it is again present inside the thallus, and you will find the antheridium to be stalked, multicellular, and they have a sterile jacket layer, which is a club shaped. Okay, so single layered. Okay, it is a single sterile jacket layer, and along with that you will find plenty of anthrozoid mother cells, antheridial mother cells, sorry, it is antheridial mother cells, which are plenty in number. Okay, so these antheridial mother cells will metamorphosize itself after the cell division, and they transform into a spindle shaped biflagellated anthrozoid. Okay, spindle shaped biflagellated anthrozoid. Along this anthrozoid has flagella, that means it is motile, and water is the medium for it to move. Okay, the next thing, moving into archegonium, you can see that they are flask shaped. Okay, flask shaped structure, which consists of swollen lower part, which is called as venter. Okay, and this particular region, that is the narrow region, is called as neck. Okay, so the venter region will consist of an egg, that is a big cell with all the reserve food material, and will have one single venter canal cell. Okay, it is called a venter canal cell, and then they will have neck canal cells. So this neck canal cells can be 4 in number, 8 in number. Then adjacent to the neck, this is again present inside the thallus, okay, inside the thallus. So they will be having 6 neck cells, okay. Neck cells means the cells in the thallus, okay. So here also there will be thalloid cells present, okay. Now this will have 2 crown cells or closing cells. So what is the speciality? As soon as this particular egg matures, the venter canal cells and the neck canal cells will disintegrate. They will disorganize themselves, and there is a mucilage which has been produced, and that mucilage will create a pressure on the neck canal cells. As soon as the pressure is created, this neck canal cells will open up. Okay, they will open up, and as they disintegrate, as these disintegrate, the liquid will cause pressure and will open up this neck canal cell. And this crown cell, as it is open, this mucilage will be present outside. This mucilage will attract the anthrozoid. Once they are attracted because of the content in the mucilage, the anthrozoid will swim, okay, and reach the egg. So once they reach the egg, they fuse, okay, they fuse. The anthrozoid will fuse with the egg and they will result in zygote. This zygote will stay inside the archegonium for quite a lot of time and will metamorphosize or develop into the sporophyte. This sporophyte is the diploid, that is the gametophytic. This particular part which we talk about, the thallus, the sex organs, etc., is called as gametophytic life cycle, and the other one is the sporophytic life cycle. So in the gametophytic life cycle, the bryophytes will show haploid condition. In the sporophytic condition, they show diploid condition, and the zygote will develop into capsule, okay, will develop into sporophyte, okay. They develop into sporophyte when they are still inside the archegonium, they develop. So the sporophyte consists of 3 regions. The first one is foot, second one is seta, and the third one is capsule, okay. So foot is the one which is holding on to the thallus and it is helping in anchorage. Seta is the one which is connecting the foot and the capsule, and this foot will even take nutrition from the thallus or the gametophyte, and that nutrition is transferred into transferred into capsule through seta. This capsule will consist of spores, which is homosporous in nature, and they are the one which is gonna grow into a new bryophyte plant once they are dehisced. Once this is burst open, all the spores are released. These spores, when they find a suitable substratum, they grow into a new individual. Thank you.